Thanks for having me here. It's always great to give a talk at Stanford, even virtually. I was a graduate of Stanford from my PhD back in 2011 or so. It holds a special place in my heart. And I'm excited to talk to you about what I'm doing here at Tempus. But first, obviously, I want to orient everybody on how I think about the world and how I got to Tempus. Before Tempus, I was a professor for many years at Mount Sinai School of Medicine in New York City. Joined Tempus about 1.5 years ago. And I'm interested in what the symposium is about, of course, is precision medicine. And obviously one of the core concepts of precision medicine is that, concepts of human disease are rooted in symptoms and anatomy still today, right? Still today, when we go to the hospital, we see that the hospital is organized around a symptoms and anatomy based sort of ontology or taxonomy, if you will. Pharmas are still largely organized in this way. And we think about organs and when organs go bad and how we treat them. And I want to, being a patient has actually made me really interested in precision medicine. Interestingly enough, it's not just a purely, academic interest for me. So I have Crohn's disease, and it's a picture of me in the hospital there. And even though I have a somber look on my face, you shouldn't feel bad for me, because when that picture was taken, I was actually loaded up on morphine. So I feel fantastic, even though I look a little serious in that picture. But one of the things that happens when you have Crohn's disease is that you get cutaneous manifestations of Crohn's disease, right? So Crohn's disease, for those that you don't know, is an immune disorder of the small intestine, largely, usually at the terminal ileum in the large intestine. Some people say it's an autoimmune disease, maybe is, maybe isn't, but you get these skin manifestations. And one thing that happens to me before I get a flare of my Crohn's that sends me to the hospital, like you see on that picture, is that I would get a rash across my chest or on my arms, like you see in that picture. And when I went to my GI doctor in New York at the time, who was a key opinion leader in Crohn's disease, I said, well, what do you think about this? This is interesting. My my skin seems to be reacting to my gut. It's almost like a sentinel, it's reacting to my gut. And, what's really interesting is the biology shared here, right? It's like, it's kind of gross to think about it this way, but your gut is your inside skin, right? If you really think about that. And obviously we know from molecular studies now, there's a microbiome, there's a barrier function. A lot of the genetics are shared between psoriasis and Crohn's, for example. So I asked my GI doctor, I said, well, what do you think about this? Isn't this interesting that my skin is reacting in this way? And he goes, oh yeah, we know about that. It's in the books, in the medical books. And I said, well, what do you think about that? And he told me, well, I think you should go see a dermatologist. So I hung my head and went to my dermatology doctor and gave the same spiel about how isn't it interesting that these skin diseases and gut seems to be connected. What's the connection there? Isn't that interesting? And I asked my dermatologist, what do you think about that? And my dermatologist said, well, I think you should put steroids on it and go back and see your GI doctor. So it's quite depressing, but it was a stark sort of firsthand experience about how medicine is siloed in such a way that it's rare that we get enough curiosity and interest to sort of figure out how these things are, even though we know they're connected symptomatically, how these things connected at the molecular level. And one of my sort of fundamental beliefs is that every disease is a systemic disease. What do I mean by that? Is that, of course, we have bodies made up of cells organized into tissues organized into organs. Inside of a cell, we have obviously DNA, RNA, metabolite networks, protein networks. We have tissue to tissue, cell to cell communication. We have organ to organ communication. And of course, the environment interacts with all these systems on a multi-scale way. So to me personally, it seems ridiculous to look at a brain disease, for example, and say, oh, that's just a brain disease. I would argue every disease is a systemic disease, and from the perspective that it's connected to the bigger body system. So how do we understand every disease from sort of the systems anatomy, systems medicine perspective? And I was a little nostalgic, since I'm talking to Stanford. I wanted to pull up a paper I published back when I was a grad student at Stanford a long time ago. My PhD advisor at the time was Atul Butte, who is no longer at Stanford. He's actually now at UCSF. But he was back at Stanford at the time. And to answer some of these questions early on, we realized there's an opportunity with some of the large emerging molecular databases that were being published. For example, GWAS studies, gene expression studies. So what we did is we pulled a bunch of data down from the Gene Expression Omnibus from NCBI and cleaned it all up and annotated it with clinical terms and things like that. But we're able to, for the first time, this many years ago now, to ask a very basic question. Does the disease taxonomy at the molecular level, in this case, characterized by shared perturbations at the RNA or pathway level. And if two diseases are connected here, it means that at a transcriptome-wide level, they share more commonality in terms of the genes and pathways being disrupted, more than you'd expect by random chance. Does that molecular taxonomy look anything like the symptoms and anatomy-based taxonomy of disease? I apologize, I got to shut something down here. It's annoying me. And so what we found, obviously, in this paper was that you find diseases that we think are very dissimilar, like Alzheimer's disease being connected to actinic keratosis. And in fact, there have been a number of papers now that show many commonalities between skin and brain diseases. And maybe if you knew developmental biology, you wouldn't find that to be very surprising, because in embryonic development, obviously, we have the brain and the skin are sort of together at one point in a structure called the neuroectoderm. And it's possible that even in these differentiated tissue states, that there's still some common molecular characteristics that bind these tissues together. So that was just taking, again, gross representations of tissue using transcriptomic data, very crude data, from our perspective today, 10 years later. But one thing I did when I became a professor at Mount Sinai was to wonder, could we apply the same data-driven approach to sort of redefine disease in this sort of unsupervised way at the patient level. So I published a paper in Science Translational Medicine, was lucky enough to get it on the cover there and get a nice write-up from Francis Collins about this paper. But when I got to Mount Sinai School of Medicine in New York, we have access to huge amounts of patients with electronic health record data. They have 100,000 patients that are sequenced or genotyped, not sequenced, but genotyped at the time. So then I asked the question at the patient level, and what I did in this case is I didn't take molecular data on patients. I took phenotypic data on patients, but objective phenotypic data such as lab results, height, weight, anything that was in their any objective measurement that was in their clinical record. And I basically represented each patient by all their sort of this high dimensional clinical phenotype data that I was able to get at that time in the electronic health records. And then I used a different type of clustering algorithm, actually invented by Gunnar Carlson, who's I think emeritus professor from Stanford, the algebraic topology method. But asked the same question, at the patient level now, what patients look like this patient, look like this patient? And what you see in this map is actually basically a 2D representation of this high dimensional clinical phenotype space where every dot is a patient or a group of patients who are similar across this high dimensional clinical phenotype space. And again, we ignored whatever diagnosis codes were given to the patient because we wanted to let the raw data, sort of lab values and things like that, drive the topology and not be influenced by the diagnosis codes. And what you see here with this coloring is we then went back and asked, where are the patients that have the sort of traditional phenotype of type 2 diabetes? We highlighted the map, and what you can see is there's sort of subclusters all over this map. So if you actually zoom in on just the diabetes patients alone and rerun the same algorithm, you find that this unsupervised clustering method clusters these traditional type 2 diabetic patients into 3 distinct clusters: type 2, type 3, type 4 diabetes. So that's there's only type 1 and type 2 diabetes, obviously. But the provocative idea from this analysis is, are there type 2, type 3, and type 4 diabetes? Well, you can cluster data all day long and get cool clusters and cool figures, but who cares, obviously, if it doesn't mean anything clinically. So we had longitudinal EHR data on these patients. So the first question we could ask was, are there any interesting clinical features that differentiate these data-driven type 2 diabetes clusters? We found that this group here at the top had increased risk of stroke and heart attack above a typical type 2 diabetic. Now, all diabetics generally have increased risk of stroke and heart attack, but if you were in the subgroup, you had increased risk above and beyond the typical type 2 diabetic. This group on the bottom had increased risk of cancer. It's interesting because all type 2 diabetics have increased risk of cancer. But if you're in this subgroup, you had increased risk above and beyond the background of type 2 diabetics. And this group was sort of a weird immune subgroup here. So I would say that's mildly interesting, right? Because it's retrospective analysis. So it kind of just hints at that these are potentially clinically interesting or even biologically interesting subgroups. But we had also whole genome genotyping on these patients, and the genetic data was kept out separate from the clustering. And then what we did after the patients were clustered was that we ran sort of genome-wide association studies on each one of these clusters. And what was really interesting was if you looked at the SNPs that clustered in this top group, they were heavily enriched in pathways related to vascular endothelial function, ion channel function of the heart, and things like that. So now the genetics was kept completely out of this clustering step. So it's hard to explain away the fact that the molecular characteristics of these patients matched this sort of data-driven phenotype cluster. And the same thing with cancer. The cancer patients had many more enriched oncogenes and things like that you'd expect from random chance. So we had this sort of independent dimension of genetic factors matching this. And it's sort of like we did a reverse GWAS, right? So instead of assuming we understood the phenotype, we actually did a data-driven exploration of the phenotype and let the phenotype, high complex phenotypic space sort itself out, and then did our genome-wide association study. So again, this paper was published in 2015, that's a long time ago. And this was conducted using only sort of the top level measurements, like the exact lab values and things like that. Then shortly thereafter, we realized, as many people did, that there was interesting opportunities with deep feature learning, and where we could say, hey, is there information latent or hidden, below this high dimensional clinical phenotype data that we could use to make predictions without explicitly using just the lab values. And it's sort of the same idea behind the first big paper that came out on deep learning, which was the Google finding cats in videos, right? If you remember that a long time ago, where there was a BBC article about Google finding cats in videos, and people wondered, well, what's the big deal? And the big deal is, of course, Google's able to take a set of millions of cat videos and millions of videos, with cats, millions of videos without cats. And instead of having humans label samples and say, oh, here's the cat, here's the I'm going to circle its ears, circle its eyes, and having human experts label those videos, they're able to let an unsupervised algorithm just learn what a cat looks like purely from data and be able to identify a cat on its own. So same idea here with the question was, could we use the unsupervised deep feature learning and for feature representation to learn models of disease? And we published this paper called Deep Patient, where we showed, in fact, that we could use these sort of unsupervised deep feature representations of patients and, for example, predict who is going to get schizophrenia in the next 90 days, who's going to get diabetes in the next 90 days. And we didn't have to explicitly predetermine, we didn't have to speak, for example, with a diabetes expert and get expert-driven features of that model, right? And we could also take advantage of the latent information hidden in that sort of, if you will, in that high-dimensional phenotype space. And I think the next frontier of all this, we published a paper on this. It's in bioRxiv. I got to get around to publishing it. On now extrapolating this type of analysis to the longitudinal dimension, right? So the limitation of that earlier diabetes paper was that it was a cross-sectional representation. So now, sort of combining things like the deep feature learning with the sort of data-driven phenotype clustering, we published a paper that sort of applies this in longitudinal time, right? Because the longitudinal trajectory of a patient itself is an interesting phenotype, right? So it's sort of the next, and other people are working on this as well, the next level of this type of analysis, especially with diseases like type 2 diabetes that progress over time, is to do this longitudinal, really understand how patients, move into and out of subclusters, for example, in longitudinal time, and what drugs or environmental factors might drive that. So, but why I really love all this data-driven stuff is not just because you get interesting papers and cool figures, and you get to use all kinds of cool jargony words that make you sound smart, but it's really that it's necessary in biomedical discovery, because in my opinion, biomedical discovery really suffers from what I would say is the streetlight effect, which is exemplified by this cartoon here, right? Where this man is looking for the quarter he dropped, and the police officer is saying, "Oh, did you drop it here?" And he goes, "No, actually, I dropped it down the street." And then the reason the person's looking there is because that's where the light is shining, right? And I think this happens in biology a lot. And in biological or biomedical discovery, is we tend to look where the light's been shining. And if you don't really understand that streetlight effect cartoon I just showed, maybe you'll understand this quote. Nobody really expects to see Donald Rumsfeld in a scientific presentation, but he said a quote that he sort of gets made fun of for, but I think is a great quote, where he said, there are known knowns and there are known unknowns, and there's unknown unknowns, the things we don't know, we don't know. And that's really what we struggle with in biomedical discovery, and for which these sort of unsupervised or data-driven methods can really help us with, is discovering these unknown unknowns. I think one good example of this from my own work was a study we published right before I had left, academia, where we took a very large multi-scale data set on Alzheimer's disease. It's called the data produced by the Accelerating Medicines Partnership for Alzheimer's Disease, and applied similar types of data-driven approaches to really try to understand what was different between Alzheimer's brains and healthy brains. But what we were actually looking for was drug targets. And I like to highlight this because it's one of my favorite quotes I've ever had published. which is I went looking for drugs and all I found were these stupid viruses. And I'll explain what I mean by that. Is again, we were trying to figure out how do we analyze sort of the totality of all these multi-omics data that were being generated on Alzheimer's brains. And we have a whole series of sort of network-based, multi-scale network-based methodologies for doing that. And what we did is we built these sort of multi-scale data networks from healthy brains and Alzheimer's brains, and we let this algorithms tell us what was different. How are these data networks rewired between healthy brains and Alzheimer's brains? And what we're hoping to find was drug targets, novel drug targets we could start investigating. But what we found instead was viral networks that had viruses at the center of them, gene regulatory networks that had viruses at the center of them. Not at all what we were looking for. But it turned out to be really interesting because it highlighted some novel, at least innate immune biology around potentially having viruses mediate the pathology of Alzheimer's disease. So this was a super unpopular paper with all of the traditional Alzheimer's researchers. They hated it. I got all kinds of hate mail. But slowly but surely, more and more papers are being published that add additional weight to this, at least the innate immune biology around viruses, antiviral biology and its contributions to Alzheimer's disease, so much that our paper and a few other papers convinced the NIH to actually open up a whole sort of line of funding to fund research in this area. And there's all kinds of very exciting work coming out of this. But again, it's one of my favorite examples of how data-driven approaches shine the light on an area that we really weren't looking and leads to novel discoveries. So that's my background in how I think about research and the type of research I'm interested in, but I'm no longer in academia. So why am I not in academia anymore? As someone who's really interested in precision medicine, and I had a great position at Mount Sinai. I mean, Mount Sinai had 3 million patients. We had a huge biobank with now over 100,000 patients genotyped. They're now being sequenced with exomes. That seems like a big sandbox, right, to play in. But one of my public service announcements for this this talk here is that I'll say it. It's fun to say it like Yoda. The precision medicine war's begun. They have, and what do I mean by that? Is that there are entities like Tempus and others now that are getting spun up that are going to create Google scale companies around precision medicine. And so precision medicine used to be, and there's still lots of precision medicine programs like that. It's like, oh, well, we'll buy a couple sequencers for our academic medical center. We'll start developing our own panels or start sequencing our patients, and we're doing precision medicine. But that's sort of like, the boutique, small batch made in Brooklyn, precision medicine. We've moved well beyond that. I don't think a lot of people have realized, but really, again, massive soon to be Google scale companies have spun up that are playing in this space. And we've really entered a whole new phase of precision medicine and getting it to scale. So I work at Tempus. I'm obviously biased towards Tempus, but Tempus is one of these companies that's playing in this space. And there's a couple of things that were really interesting about Tempus, which has started. So Tempus started about 5 years ago. Was started by Eric Lefkowski, whose wife got breast cancer. He was a technology investor and entrepreneur, and he realized the deficiencies around data in the health system. Founded Tempus. Now we've raised 1.1 billion. We've got about 1,800 people that work in various cities for us. And I think we've become one of the largest sequencers of cancer patients, at least, and we're expanding vastly beyond that. We'll sequence a couple 100,000, cancer samples this year. But what was interesting about Tempus that I'd never seen before, it was the first precision medicine company operating at scale that was totally committed to multimodal data. So there's lots of precision medicine companies that are doing things like targeted DNA, panel sequencing and things like that, which is super boring to a systems person like me. And what you'll soon see is that Tempus, in my opinion, is the only precision medicine company that is a data-first precision medicine company. And what do I mean by that? What I mean by that is when we get a sample in our hands, the first thing we think about is how much data can we generate from this sample without completely going crazy. There's a lot of testing companies that say, oh, we're data companies. But when you look at their business model and how they operate, they're actually much closer to LabCorp and Quest than they are to actually being a data company, because all they're concerned about is cheap tests at high volume. So Tempus has a number of interesting aspects to its infrastructure. So we're connected into over 200 different health care providers, everything from academic medical centers to community hospitals. We have real-time data pipelines that connect to EHRs, data warehouses. We have our own sequencing facility. We have something like, I don't know, 16 NovaSeqs or something like that, just cranking away all the time. So we pull this real time. We pull in clinical data. We pull in. We have generate huge amounts of sequencing data, and we have a whole machine to sort of crunch on this data and do all kinds of interesting things with it. As I mentioned, we're working with 80 of the top 100 hospitals in North America. We have over, I think, 200 hospitals in our client base. But quite simply, Tempus is in the business of developing smart tests. So everybody's excited to bring AI to healthcare, right? So I'm one of those people. And the question is, how do you bring AI to healthcare? So one way you can do it is to bring, make the EHR smart, right? And we all know the challenges with that. We have sort of, the Epics and Cerners of the world, and anyone who works with those systems knows that they're far from being smart in terms of embedding true AI into those systems. So that's a difficult path. The other thing you can do is build an AI system and try to bring the physician, a separate AI system, and bring the clinicians there. But that's not an easy path, because clinicians don't want to be taken out of their existing workflows. Healthcare systems are already committed to things like Epic and Cerners. So that's a difficult path. And that road is littered with many bodies in terms of companies that have failed to try to do that. So the basic concept is we develop smart tests. So we're bringing AI to medicine by developing smart tests. And what I mean by that is physicians interact with clinical tests all day long, and things like genetic tests, things like laboratory test results. So how do we actually contextualize those tests and make those tests smart? So we're just making something they interact with every day smart. So I'll walk through that in a second. And the way we do that, again, is by generating modal clinical data. So let me walk you through what happens when a physician orders a Tempus cancer test, so you can really understand what I'm talking about here. So if someone orders our cancer test called XT, we have many other tests. We have liquid biopsies, we have things like that. But let's say they order our XT test, which is for our solid tumor. The tumor will come in. Let's say the solid tumor section. And we'll also get normal blood, which is strangely unusual. Many companies don't even get the normal blood, so we can compare against the germline. But we'll do a targeted DNA sequencing panel of about 600 some genes. That's not that interesting. Lots of people do that. We also generate full transcriptome on every sample that comes in the door. We don't get reimbursed for that at all. But again, I told you, Tempus is a data-first company. So when we get a sample on our hands, we first think, what valuable data can we generate from a sample? So from day one, we've been generating full transcriptome on every cancer patient that comes in the door. We now have the biggest cancer transcriptome database in the world. If you're familiar with the Cancer Genome Atlas, it's not even close to Tempus. The Cancer Genome Atlas is a tiny, tiny data set compared to what Tempus has built in the last 5 years. We also have our own pathology lab. So we also section and stain and do high resolution imaging of pathology imaging of the tumors. We also integrated into the EHR or the clinical data warehouse. We pull in that patient's clinical data, we structure it. We have NLP and things like that, but we also have about 200 human curators that check the quality of the NLP. So there's humans in the loop of that step. We structure that clinical data, and then we actually generate a report, a smart test report for that patient. And one of the things we do, which seems like everything a test should be able to do, which is sort of depressing about the state of the art of the whole industry. But so, for example, if a patient has a mutation that suggests they should be on a certain drug, but we know from their clinical history that we pulled and structured that they were on that drug and failed it, we will dynamically update our report to reflect the fact that they already tried that drug and failed it, even though they have a mutation that suggests that they should take it. That should be table stakes, you would imagine, for every cancer test, but most tests are dumb and they're not able to do that. So we generate the smart report that integrates imaging data, integrates transcriptomic data, integrates DNA data, and more, and we send that back to the clinician. So we're not just a company that's taking data, we're taking data, synthesizing it for that patient, and pushing it back to the provider and delivering a report. And one of the things I love about Tempus and helps me sleep well at night is we generate huge amounts of molecular and clinical data on patients. And when we not only deliver the report back to the health system, we deliver back the data, and the data is theirs. Right, so the data is theirs to keep. We keep a copy, they get a copy, but we don't put any restrictions on that clinician's ability or that health system's ability to use that data for whatever they want. So, and one of the other types of tests we build is what we call algos or digital tests. So Tempus is a bit unusual compared to a typical diagnostics company, right? So a typical diagnostics company, if they wanted to come up with a diagnostic, they would say, oh, well, we want to come up with a biomarker for, let's say, who's going to respond to some drug. And it's going to be RNA based. So a typical program around that would use whole transcriptome as their discovery set, right? And say, oh, well, let's see if we can discover from whole transcriptome sequencing in our discovery cohort a gene signature that correlates with drug response. And let's say in that example, they find like a 15 gene signature that correlates with drug response. A typical diagnostic company would then say, oh, let's use nanostring or something like that and go to market with this 15 gene test. That's not what Tempus does at all. Tempus wants to go to market with the broadest possible test. So if we're going to do anything in diagnostics for transcriptome, let's just go to market with both transcriptome by default. Because as you learn new gene signatures, and if we're running transcriptome by default in every patient, that means that any future discovery for a new gene signature is overlaid digitally onto the RNA that's already been generated for that patient, right? So we always want to go to market with the broadest possible test by default. So all future insights are digital overlays, or algos as we call them, on the patient data. So HRD, or homologous recombination deficiency, is one such test we have that's basically just a digital test that gets run on top of data we're already collecting from the patient. One of the other key features of the Tempus ecosystem is we have a national clinical trials network. So again, Tempus is really a company that's optimized around the network effects of our data and the data we generate, and then all the different ways we can use that data to develop novel tests, obviously to deliver patient-specific insights, but also to match patients to trials. And we just don't hand that patient off to another health system, or I'm sorry, another company like IQVIA or something like that, or other, maybe that's not a great example, but another clinical trial company. We actually built a nationwide clinical trial network around the United States. We have 4,000 oncologists in this network, and we're also taking in data from these sites. But as patients get matched into our system, they actually then get matched into our clinical trial network that we built around the country, so that they can match somewhere close to where they live. So we have a rapid activation. This is called the Time Network. And what I want to skip to is this is the most impressive part, is now that we've built this national network, if we match a patient, we can activate a single patient clinical trial within 10 days, or typically 10 days of matching that patient. So again, the whole network is already spun up so that everybody's on a common IRB, a common rate sheet, et cetera. So if a patient matches in rural Oklahoma, we probably have a time site close to where that patient lives, and we will rapidly activate a single patient clinical trial. So we typically go from a patient matching a study to getting their first dose in 10 days or 10 days or less, instead of driving 100 miles to the closest academic medical center. So, and we also have a suite of, in addition to all the data we're generating, we have a suite of tools to interrogate this data, to do things like I showed you with the type 2 diabetes data set around cohorts, cutting cohorts, exploring cohorts. You can sort of say, put your patient in the middle if you're a clinical, if you're treating someone clinically, and say which patients in the whole Tempus data universe look like my patient, and then start playing around with that data to say, oh, well, do patients who look like my patient, that they do well on PARP inhibitors? You can start asking questions like that. And we're not in the business of selling software. This is just all sort of the software we built around our data to help people use our data who interact with us as customers. So that's cohort. And then we also have Lens, which is sort of a power user platform for people doing research on our data. The other thing I forgot to mention in my cancer example is not only do we, like I said, do DNA sequencing, do transcriptome sequencing, do digital pathology, do clinical data structuring, we also have an organoid facility. So for every patient we test, we try to grow and store a patient-derived organoid. As far as we know, we have the largest cancer organoid bank in the country, perhaps the world. Now, the idea here is obviously for every patient we test, we have their clinical data and their molecular data generated from their tumor, and an organoid that we can grow and use for drug screening that will have all that rich, sort of in vivo data from the patient and their clinical data attached to it. So, and we have a huge effort and a huge lab to screen these patient-specific organoids, work with drug companies, and with eventually plans of developing patient-specific tests around organoids as well. So again, it's all about data. We deliver high-quality tests with great insights, but it's really, like I mentioned, a data-first company. I've also made some pretty large investments in systems immunology and collecting immunological data. So as I mentioned, we do whole transcriptome, but we don't just use off-the-shelf, sort of transcriptome kits or libraries. We've developed our own RNA or transcriptomics panel. One of the things we added to the version 2 of our panel is TCR-BCR probes, so typical of what you would get with like an adaptive biotechnologies. So again, for every patient that's clinically tested at Tempus, we'll run this transcriptome panel, and now we have a large and growing library of novel TCR-BCR receptor sequences, which we can do all kinds of interesting things with, obviously from supporting cell therapy to drug discovery, et cetera. So again, Tempus is all about maximizing data. One of the other nice benefits of Tempus is because we have such large testing volume, as we have a huge number of samples coming in. So this is an example where we grabbed some non-small cell lung cancer patients that had some responders and non-responders to immune therapy. We did some site-seq and single-cell sequencing, and we found a novel T-cell, cytotoxic T-cell signature from the single-cell data that we thought was tumor killing. And the beauty is, okay, well, great, we have a signature. What do we do with it? Well, we have a massive database. Like I said, we run transcriptome by default on every patient. So we have a huge real-world database of RNA data connected to clinical data, connected to outcomes. So the beauty of our system is once we discover that signature in our lab, we can go turn around right away to our growing massive data set and try to validate that signature on real patient data. And in this case, we're able to, this cytotoxic T-cell signature showed a difference in survival for non-small cell lung cancer patients undergoing immune therapy. So this is the power of this sort of closed loop data network where we can generate these types of signatures rapidly, but then we can quickly validate on our growing real world multi-scale, multimodal data set. One of the really interesting things you can do when you have this sort of rich multimodal data, particularly imaging and histopathology connected to the molecular data. This is work that our data science team is doing, is you can predict all kinds of molecular features off the imaging, right? So we have a huge program, a number of products in development, and we've already published some of these where we can actually predict, again, because we have all this matched data on the same patients. We have several deep learning algorithms that can predict molecular features such as MSI status, things such as transcriptomic pathway activity, et cetera, just off of H&E slides. And again, now that will feed into our clinical trials network, because now instead of sequencing patients at these clinical trial sites, you might be able to just scan a slide and infer RNA pathway activities or DNA features such as microsatellite instability from the image alone without ever having to run sequencing. And this is how this multimodal data network starts to enable these types of applications and feeds into our other businesses. I'm sorry for my cat in the background making noise. This is an example of our algorithm that calls microsatellite instability purely from the H&E alone. We have another one that actually can call transcriptomic-based consensus molecular subtypes for colorectal cancer, again, purely from the slide alone, but this is only enabled by this large scale of connected molecular, clinical, and H&E data. And I'll wrap up here. I've lost track of time, so I'm sorry if I'm going over, but Tempus is not just a sequencing company. So as part of our algos business, we want to be able to get new insights off of tests that are being run as part of everyday care and make these tests smarter. One area we're doing that is actually in cardiology. So we started to expand into cardiology as another vertical, where we're running deep learning models on top of ECG traces, and we've published a couple of these papers now, where we can predict things like mortality, AFib, acute coronary syndrome, stroke, et cetera, just from a single ECG trace. Now, we're not the only ones doing this, but we're definitely the only ones taking this to the FDA and getting them, with the aims of getting them approved as a software, as a medical device, and rolling them out across all of our healthcare system providers at scale. So the idea behind this type of work is we put an ECG in, and instead of in this deep learning model, and instead of trying to diagnose something in the episode of care, we're trying to predict something that's going to happen in the future. So the punchline here is you can see from this graph on the left here, is that there are ECGs that were predicted to be or clinically sort of annotated to be normal, but our algorithm predicted them to be high risk for AFib. And this is in collaboration with Geisinger, and we have about 700,000 patients or so with ECGs and full-length clinical data. Geisinger, they have very deep clinical data, and the algorithm is able to find abnormal or AFib ECGs, even though they're clinically annotated to be normal. We also have shown that this is predictive of stroke, future stroke development in more clinically important events like that. We also are working on sort of interpretability of these deep learning models, which is always a challenge with sort of deep learning based diagnostics, to actually paint, when we make a clinical prediction about AFib or stroke, to actually paint the ECG trace to sort of highlight the areas of the ECG for the clinician that are sort of driving the prediction. And like I said, we have a whole suite of these ECG algorithms that we're taking to the FDA. The software is a medical device, and the plan is to have something, we have a handful now. We hope to have 20 or 30, in a couple of years of these different ECG algos that can be run. So as I mentioned, the Tempus, sort of connecting back to my earlier slide, Tempus started in cancer. We're expanding into cardiology, and we're doing a lot of other work in cardiology I can't talk about. We've done a lot of COVID testing. We're expanding our footprint in infectious disease. And we actually have a whole neuro vertical I wasn't able to talk about. IL-6 is connected to all those different disease areas, such as neurology and numerous papers, but there's still so much we don't know about that cross connection between those diseases. So the hope here is by building up these sort of Tempus scale data sets in cardiology and neurology and other areas that we're going to start to really fill out our understanding of these sort of molecular connections between these diseases. So that was it. So sorry if I went over time.
Oh yeah, no worries. No, that was really interesting, and yeah, it was great to see that. I guess everybody has their own idea about scale, so this kind of it seemed like kind of blew out of everything I've been seeing out of the water a bit, which is exciting. Yeah, so I guess just ask you a couple of questions I think we have time for. You mentioned the spotlight effect, and you mentioned really prioritizing getting as much data as possible. So how do you set a limit to how much you pull from any given sample? Like, are you the one that decides, okay, we're going to run this, and that's within our budget, or how do you let the algorithm decide for you? Like, how constrained are you?
Yeah, it's a good question. I apologize, my connection's not too fast here. So, it's really there's some thought that goes into it in terms of what dimensions of data we think will be useful, if not today, in the near future, right? So I love transcriptomics, so it's sort of depressing to me that transcriptomics still isn't used that much in cancer. Even myself and other people have published papers on obviously it's a much more dynamic molecule, right, that tells us a lot more about what's going on inside the tumor, for example. But that there's enough feasibility there to know how useful that is in cancer, and we hope it's imminent in clinical care, and we sort of trying to help usher that in by collecting it. And same with we have a product under development for whole genome methylation. As I start to collecting that by default, for example, even though that won't be used clinically for some time, but to try to build up that data set.